I now have the opportunity to introduce a longtime colleague from the National Institute of Environmental Health Sciences, and this is Dr. Ray Tice. He's going to be speaking today about the in vitro and high throughput screening, HTS assays, and we appreciate you being here. The floor is yours, Ray.
Thanks. I really welcome the opportunity to speak today, and I have to admit, after the talk yesterday afternoon about the microbiome, I have to say that my microbiome also welcomes being here in Italy. One of the things I'm going to talk about is what we call the Tox 21 program, and let me start, not with that button, that's for sure. All right, so the formation of the Tox 21 community formally was started with a 5-year memorandum of understanding on high-throughput screening, toxicity pathway profiling, biological interpretation of findings on February 14, 2008. You notice it was signed by Francis Collins, who's now the director of NIH at the United States, Sam Wilson representing NIEHS, and George Gray representing EPA. In 2010, we expanded it excuse me, to include the Food and Drug Administration in order to hopefully have access to drug information and human data. And then in September of this last month, it's been signed again for a new 5-year MOU by what's now the NCATS, NIEHS, EPA, and FDA. This is one of the few examples of government interagency work at the U.S. that seems to be working and will continue to work as far as I can tell. What are the goals? They're very straightforward. One is to identify patterns of compound-induced biological response in order to characterize toxicity disease pathways, facilitate cross-species extrapolation, model low-dose extrapolation. The initial and only goal was to be able to prioritize compounds for more extensive toxicological testing. This is driven by the fact that over the history of the National Toxicology Program, since about 1979 or so, it's only been able to evaluate completely maybe less than 1,000 compounds. And at the same time, more and more compounds being pushed out into the environment. Ultimately, we'd like to be able to use the data to develop predictive models for biological response in humans. So we've had 3 phases. The first phase was pretty straightforward. We called it proof of principle. It actually started in 2005 before the existence of the Memorandum of Understanding. And there's 2 components to this. EPA had what they call a ToxCast program, which focused on a small number of compounds, about 320, mostly pesticide actives, and some endocrine active compounds to be evaluated in about 550 assays. And all that data is available on EPA Actor. At the same time, we sent up to the NIH Chemical Genomics Center 1,408 compounds. EPA provided 1,462, and we ran them in quantitative high-throughput screens that I'll describe in more detail across about 77 predominantly cell-based reporter gene endpoints. And all these data have been made public versus either in PubChem or in a database that's housed within the National Toxicology Program called SEBS for Chemical Effects in Biological Systems. Phase 2, which started in about 2011 and continues currently, for ToxCast involved about 700 compounds being screened in more than 700 assays and a subset of 1,000 compounds being screened in endocrine disruptor. For NCATS, Quantitative High Throughput Screening Program, we put together what we call the 10K library, or 10,000 compounds, which I'll describe in more detail on the next slide. And then we selected a bunch of assays to focus that library testing on, and the assays focus on nuclear receptor activation or inhibition, induction of cellular stress response pathways, and we managed to also include a project to try and characterize human variability in response that I'll describe. All of this data is being made public versus the ComTox website at EPA. They have the data either downloadable or on a dashboard. It's also on PubChem, and then again, the data is also available on SEBS, and we've been making the data available to anybody who wants to download it in a special site. So what's in the 10K library? So it's actually, this is a slide on version 1. Version 2 has been altered a little bit because the drug library provided by NCATS expired and they provided a second drug library. But basically the library is pretty much the same. So what you have here is, on the left is the ToxCast phase 1 and 2 compounds. The rest of the compounds of interest to EPA. The ones on the right show you the compounds that are of interest to the National Toxicology Program, including a set of formulated mixtures. And NCATS provided a set of compounds that came out of their drug library. These are drugs that were on the market in Europe, Canada, United States, and Japan. There are 8,307 unique compounds, but a total of 12,000 wells were occupied by compounds. And you can see by the Venn diagram, there's a number of compounds in duplicate and triplicate. We also included 88 compounds single-sourced in duplicate on each plate to look at technical variability. So there was lots of redundancy built into this process. In addition to that, we've done chemical analysis on all of the compounds in the library. This effort actually took more years than it took to do the biological sampling because of the difficulty in testing that number of chemicals. But the compound identity and structures are available on the website that's indicated at the bottom. The library actually contains about 1,700 food-relevant compounds, direct additives, indirect additives, food contact, substances, and pesticides. These were not picked because they were considered food-relevant, but they're part of the library as it exists. Basically, what's called quantitative high-throughput screening, which is conducted at NCATS. All the assays are homogeneous. That means we can't do any wash steps. We just basically add, mix, and read. The library is DMSO-soluble compounds. We weren't testing water-soluble compounds. It's 1536 well plate format, 15-point concentration curves. Each plate is a separate concentration. Typically, the concentrations range from 15 nanomolar to 92 micromolar, 5 microliters, 1,000 to 3,000 cells, and we run each assay 3 times, generating on the order of 36,000 concentration response curves per assay. That's a little bit hard for any one individual to look through and look at every single concentration response curve and make sense of it. So we're talking about big data, but it was extremely important to run this in 3 runs. By doing that and changing the well locations, we were able to identify both systematic and non-systematic errors associated with each assay, and those are flags that are attached to the assay results. This is a list of the nuclear receptor and related assays. You can go through and read them. The one thing that I think was of special interest, we did both the estrogen receptor and the androgen receptor in both the partial ligand binding domain, which is used by pharma, and also the full receptor. And not surprising, there are chemicals that are active in one of those assays but not active in the other. So basically that tells us you need both kinds of information. But it just gives you an idea of what's been done. The ones in bold are assays that have been completed and have already been made public. The other set was stress response pathway assays. The reason for picking this was the thought that if a chemical induces a stress response over a concentration range that might be relevant, it can't be good for you. So we went through and picked up endoplasmic reticulum stress, genotoxicity, heat shock, hypoxia, inflammation, oxidative stress, and then multiple stressors, including mitochondrial membrane potential, which actually turned out to be one of the assays with the biggest number of hits. We have 2 graphical user interfaces that have been developed by one of the staff, Ray Washi, as an employee as the National Toxicology Program. And if anybody's interested, I can provide the URLs later on. But this one is for data visualization, showing you an example on the right. And the next one is for compound profiling, giving you a heat map that allows you to select the data based on either the AC50 or the point of departure, or something we call weighted area under the curve, taking into account the relative potency of the different compounds. So this is something that we threw together, along with one of my colleagues, Dan Svoboda. So this is the hit rate of the 1,700 food-relevant compounds in the 10K library. We've separated them out as direct additives, indirect additives, food contact, pesticides, and direct additives, GRAS post-1999. And what I want to point out to you is that up at the top, the 1 means that these are the number of compounds, of which there's about 180 that were active in only one assay. And down here are compounds that were active across 52 assays. So that meant that they had a legitimate AC50 that was determined based on statistical analysis. And what I've added to this is to show you the compound that was active. Dodecyl gallate was the one that hit 52 assays. For the indirect additives, it was tributyl chloride. For the pesticides, it was hydromethalone, which hit 64 assays. And I won't read the compound on the bottom, but it also hit 54 assays. So those are compounds that were biologically active. It doesn't mean that poses a hazard, but they were biologically active across those assays. So one of the things, there's multiple ways of analyzing the data. You can look, say, within the estrogen receptor and rank the compounds by their potency in terms of stimulating, acting as an agonist on that particular molecule or receptor. However, Scott Auerbach and Dan Svoboda at the HTS have developed another approach for looking at it. You can call that nearest neighbor analysis or maybe even read across. But what they've done is they've cross-correlated all of the data across all of the assays. Then they look for, using a software program called Partec, they look for similarities of biological response or bioactivity networks. They then take that data, they put it on a cytoscape, and what you end up with something like this, which is very hard to read, but this is a map of all the relationships of all the compounds. The closer the two chemicals are linked to each other and showing the same bioactivity network, the closer the dots are. There are some dots on there that represent blue, and these are within the direct acting food additives that I picked out just to show you where they might be. And as an example, what I've done is pulled out one compound, which is 4,1,1,3,3-tetramethylbutylphenol. It's a direct acting food additive. Dan gave me this particular data, so what you can see is the compound name on the left, the Cas number and Pearson's correlation, or R. These are the compounds that are linked to that compound showing the same bioactivity profile across all of the assays. You can see that there was a second copy of the same compound. It showed a Pearson's correlation of 0.96, meaning it wasn't perfect, but there were some slight differences. That's one of the reasons we have redundant compounds. And then as you go down and read some of the compounds that are in there, you can recognize most of those compounds as being biologically active in the estrogen receptor. So what this compound had the biggest hit in at low micromolar concentration range for AC50 was the estrogen receptor. It was also an antagonist in the androgen receptor, and it also showed biological activity for H2AX, which is a measure of DNA damage. And then the last assay it showed activity was reducing mitochondrial membrane potential. So this is a compound that would be of interest to us for further studies, or at least going in the literature and seeing what else is known. One of the things we've been doing is to try and take into context what else is going on in terms of what we're exposed to, because we recognize that just because you have bioactivity in a particular assay, it doesn't mean that it either poses a hazard or a risk. So in working with a collaboration that was started by Barbara Wetmore and Rusty Thomas while he was at the Hamner Institute, we've been assessing the bioactivity of various fruit and vegetable juices provided by Barbara. So she's been testing them through various ToxCast assays. What I'm showing you here is the same water-soluble or the same active juices that have been taken from various fruits and vegetables listed on the left, and then looking at seeing what their biological activity is across about 9 different assays. One of the things we ran into, as you might expect, is many of these fruits and vegetable extracts are colored. So that meant that any of the assays that use fluorescence as a signal would not work because they would interfere with the fluorescent signal. So the assays we focus on use a luminescent readout. So what you can see on the right is the top 10 most active juices. The number 1 is beets. It hit across 7 of the 9 assays. It was highly potent, meaning less than 1% of it was active. It was also moderately potent or potent in the other assays as well. I noticed that garlic was number 2, and that's something that I'm very much like in the food that I eat especially, and this just points out that You're not surprised by we're exposed, as been talked about previously by Thomas, to lots of things in our diet and lots of things in our food that have bioactivity. The question is whether they pose a risk. It may pose a risk to some people under certain circumstances. I do want to mention, but I won't show any data, and this was mentioned also by Nigel Walker, who gave a presentation on the first day about NTP's efforts. We're using the high throughput screen to try and look at patterns of response for botanicals. This is a list of all the different botanicals that are going through that system. Cynthia Ryder at NTP is the lead on this, and one of the reasons for doing that is when you go out and get a botanical, they vary greatly from source and where you buy it, when it was made, who processed it. And so we can't go around and test every single botanical version of, say, golden seal root powder, of which we have 9 different versions. But by running them through the high throughput screen, we can see how they vary or how they are similar in response across the pattern of biological activity, and that can help us then decide what compound to go forward with. I do want to mention one study we did within the phase 2, which we called the 1,000 Genomes Toxicity Screening Project. This was a study that was in collaboration with Yvonne Rusin's group at that time at UNC Chapel Hill. Basically, it took 1,086 human lymphoblastoid cell lines representing 9 geographical racial groups. We screened against them 179 compounds, including 9 duplicates at 8 concentrations. The assay endpoint was cytotoxicity because that's the one we could measure when we did this. We generated 2.4 million data points, and it was correlated against 2 to 5 million SNPs. Over on the right, you can see there's 2 graphs. One shows a compound that has very little variability, another one with high variability. And so we could see how much variability there was, and then we could try and figure out what genes were responsible. This data was put out for crowdsourcing, and there's 2 papers right there, one dealing with the actual data, the Abdo paper, and then the other one dealing with the crowdsource. So phase 3 is improving on biological coverage and relevance. We recognize the limitations of doing the assays we are doing, so we're focusing on more physiologically relevant in vitro cell systems using human stem cells, cardiomyocytes, neurite outgrowth assay. We've got a lab working on 3D models, especially focusing on HEPA-RG cells. We're using more in silico models, and as we develop more data, generating QSAR models. We're testing a lot more formulated and natural mixtures to see what they do. We have increased use of alternative animal models, mostly focusing on zebrafish and C. elegans. And one thing I want to mention specifically is the development of a high-throughput transcriptomics platform that is focusing right now on human and rat, but would be developed for mouse, zebrafish, and C. elegans. So this was called the S1500, is now the S1500 Plus. We put a draft out in the scientific community of this gene set on earlier this year, and then it's now been revised to include 2,753 genes, which has also been released. And we're evaluating various technologies and platforms, and the goal is to be able to run this platform for under $25 a sample. We think we can get it down to about $10 a sample, and therefore collect data not only on time course, but also on dose, and run it against any kind of cell that you might want. We want to go away from a single endpoint assay to one that allows us to evaluate across the whole biology of human pathway space. So what is the future of toxicology? This is what I think the future is. This is where I think we need to go. One is an increased use of computational methods. As more data are generated, you're only as good as the data that you have and the extent of data. The increased use of in vitro 3D models and organoids, you'll hear more about that in the next speaker. An integrated system approach based on high content screening and omics, whether it be proteomics, metabolomics, or our focus right now is transcriptomics that can be applied to any cell type and looks across the breadth of biological space. And by having that, we think we're going to be able to better predict human health effects while taking into account sensitivity across cell types, status differentiation, development windows, and individual susceptibility. But we also think we'll allow for better extrapolation from other species because we'll know what pathways are in common and what pathways are different. I want to close with saying that Tox 21 is a collaboration of many. The people up at the top represent the Biomolecular Screening Branch and NTP colleagues. We have the National Center for Advancing Translational Science and the EPA National Center for Computational Toxicology. And as was pointed out, this is an effort that's being supported by the U.S. government. And as long as the U.S. government exists, this effort will continue. Thank you.
Thank you very much, Ray. We have the opportunity for a question or two, just for clarification. Yes, please.
Hello. If I have to consider a chemical which is intended to be added to food or feed, and I have no data so far, and if this compound is run in your assay, so how can I use these data to make an assessment of a possible risk, or can I, or is it not possible? Because I see there can be created thousands of endpoints, and how can I use it to make a decision?
Right. So one of the things we recognize, and when we started this process, we put together a diagram that said what things were easy and what things were hard, and over a 10-year time period. And the hardest thing we could identify, which actually has already taken place, is how do you use this data in making decisions? Right. So right now what we're using the data to help prioritize compounds for more extensive testing, whatever that more extensive testing might be. But however, one of the things that I will note just from the cytoscape that I showed you is that we can look at patterns of response for chemicals that we know something about. And if you picked a chemical that you knew something about based its toxicity, it's like read across, and you had a chemical in that was a food additive that showed the same pattern of response, then I would start being concerned or at least interested in that chemical because of that correlation. But it's a correlation. It is not a fact. It is not a cause, right? But if a chemical walks like a duck and quacks like a duck, then maybe it's related to a duck.
Yes, one more, please.
Jean-Loup Nefsa, thank you, Ray, for your very inspiring talk.
You're welcome.
Just a small question. In food safety, we're really interested in the intestine and the liver, of course. So we're taking into account metabolism in the liver, but also transporters in the gut and in the liver. How do you think we should address the transporters to bring that back to the physiology in that context?
Right, so recognizing the limitation, so what we've done in the TOX-21 high throughput screening program is looked at direct compounds, right? The activity of those compounds in reporter gene assays, which does not include either metabolism or transfer, right? So what we're doing is trying to do with both. We recognize those limitations. We've looked at models that allow you to predict metabolites and also allow you to predict whether things are transferred across the gut or not transferred across the gut. We've started using those. At the same time, we're trying to develop the biological system. So we have a 3D HEPA-RG system for looking at metabolism. And we're also hooking that up with the ability to look at what's a metabolite, how many metabolites are produced, and what's the dose to the target tissue. The big problem we have found is we don't know what's actually free in the medium and what's actually absorbed in the cell. And it doesn't matter what system you're talking about. So we're trying to do the same thing with zebrafish. What is the dose to the target and what is the effect that we're seeing? So, but I agree with you. That's always been the limitation, but we did not have the technology when we started to embrace those. Yeah.
Ray, thank you very much.